The next step after an extraction is a PCR. So now I'm going to perform a PCR, and these are the things you need. You need the DNA template, you need the buffer, the magnesium chloride, the DNTP, the tag polymerase, and also you need 2 primers, the forward and the reverse. So first I'm going to add the water, 12.6. 12.6 of the water. Next I'm going to add the buffer. I'm going to add 5.4 microliters of the buffer. And after I add the buffer, I'm going to add the magnesium chloride, 5.4 microliters again. Then I'm going to add 7.2 of the DNTPs. Okay, now I'll add 9 microliters of betaine. Vortex, you mixed it up a little. Okay, we'll add 1.8 microliters of the primers, of both the forward and the reverse primers. This is the reverse primer. And last but not least, I add 0.27 of the tag polymerase. And then I'll repeat this process that I just did for the next 2 cocktails that we have. I'm adding 14 microliters from the first cocktail into the first 3 tubes. And then I'll repeat the process for the next 2 cocktails, 14 microliters also. So now I'm adding 1 microliter of the DNA template into the first 3 tubes, and then I'll repeat the process with the next 2 DNA templates. 2 tubes. Then I'm going to take the last DNA template and do the same thing for the last 3 tubes. 1 microliter also. So now all the DNA template is added for each 3 of them. Okay, we'll put them in the little mini centrifuge to take all the bubbles out. Just real fast. Take them back out. We'll cover them. We'll bring them over to the machine. Just place them in a thermocycler machine. There's 3 steps to PCR. First is denaturing at 96 degrees. Then you have the annealing at 42 degrees, and then you have the elongation at 72 degrees. It takes about 3.5 hours, and it repeats itself 35 times. We'll now prepare a gel to check the PCR product. So right here we have 50 microliters of the TAE of a 1 concentration, right here. And then we'll add 0.60 of the agarose 2 of the TAE into the liquid. Okay, and once we do that, shake it up a little.
Fast.
And then we'll put it in the microwave until it has a fine, we'll heat it up until it has a fine ball. Shake it a little. You want all of it to dissolve. Okay. Now it's better. You can see the fine ball and it's all dissolved. I'm going to take it out. Then you're going to go ahead and you're going to add 2 microliters of athenium bromide. You're going to go all the way into the fluid and just release it. And you're going to shake it up. Make sure it dissolves. to dissolve, then you're going to take it and you can now pour it into here. Okay, we'll now pour it into the tray. Place the combs into the tray. When the gel hardens, we can now take off the combs, and then we can add each PCR product into the wells. It's connected to the electrophoresis, and we can go ahead and start running the products. Okay, the electrophoresis is now done. So now I'll take the gel out gently. I'll place it in here. Then I'll bring it over here to the UV trans eliminator, and I'll check for bands. Okay, now you can see that I have bands. So I'm gonna go ahead and I'm gonna cut each one of them individually, and I'm gonna place them into a 1.5 little. So I'll go ahead and take the gel off. And I'll cut them individually using a different razor each time. So I'm gonna start with the first one. Separate it from the rest and just cut around the band. I'm cutting the next band. And after I finish cutting, I just want to get around the edge closely so it's not too thick. Pick it up with the razor blade. And I'll go ahead and place it into the next two. Just carefully put it in there. Put it down and close it.